Hi everyone, this is the 13th lecture for your chapter Cell. In the last class, we discussed cytoskeleton. It included the microfilaments, intermediate filaments, and microtubules. I hope you remember all of these components because we will be using them today to study flagella and cilia. What are flagella and cilia? So they are movable protoplasmic processes. Okay, so first thing, they are movable, they can move around. And where are they coming from? Where are they arising from? So they are coming out from the protoplasm, okay, from the cytoplasm. So they are cytoplasmic processes which are movable. What function do they do? So they are helping in locomotion, which is movement from one place to another, and they also help to pass things along. Okay, so for passage of substances, to move something, they help. Okay, we shall see that when we study the functions. Okay, now if we talk about flagella, these flagella are longer. They are long. How long? So 100 to 200 micron, but they are fewer in cell. Okay, so if this is one cell, it may have 1 to 4 flagella. Okay, many protists, including what protist do you know has Euglena? Euglena has a flagella, right? Then motile algae like Chlamydomonas has flagella. Then spermatozoa of animals. Okay, spermatozoa is sperms of animals. They also have flagella for movement, right? Then bryophytes, pteridophytes, they have flagella in the spores. Okay, they are spores or they are spermatozoa, have flagella. Then choanocytes of sponges. If you remember little bit of your animal classification, try and think of sponges, and the sponge had inner lining, right? And that inner lining had collar cells called choanocytes. These choanocytes have flagella to produce water current. Okay, it generates water current. Then gastrodermal cells of coelentrates. Coelentrates are, think of, let us say, jellyfish. So in a jellyfish, in a jellyfish's stomach, you will see such kind of flagella, okay, in its gastrodermal cells. Then zoospores and gametes of thallophytes. Thallophytes are the plants which have thalloid body. It includes algae, then it includes fungus, it also includes bryophytes. Okay, so they are zoospores. Whenever you hear the word zoospore, it means the cell is moving. Okay, so zoospores and gametes of thallophytes also have flagella. Now let us talk about cilia. Compared to flagella, cilia are very small in size. Okay, they are 5 to 20 micron long, but they are numerous. Here in this picture, what you are seeing is actually a paramecium. Okay, this paramecium has these green colored cilia. Can you see how many cilia are there? Numerous, and they are small in size compared to the flagella. They occur in ciliata group of protista. Okay, in protista or in unicellular organism, you have a group called ciliata, which is all the organism, unicellular organism which have cilia. Example of which would be paramecium. Okay, then flame cell of worms. Flame cells are the excretory organs of worms. Okay, so they have cilia. Epithelium of respiratory tract, renal tubules, oviductal funnel, all of these places have cilia. Okay, both cilia and flagella are made up of the same parts. Okay, they are basically made up of the same parts. They are same in structure also. We will see what are these parts. So they are made up of the basal body. Okay, as the name suggests, it is a body or it is some organelle which is present at the base. So basal body, rootlets, again as the name suggests, what does a root do? A root fixes something, right? It anchors something. So here there are rootlets, then there is a basal plate, and then there is the shaft. Shaft is the one, the whole filament type structure that you see is called shaft in case of eukaryotes. Okay. if we were talking about bacteria or prokaryotes, that shaft would be called, what would it be called? Very good, right? It would be called a filament. So let us start with basal body. Basal body is also known as kinantosome, basal granule, or blepharoplast. Okay, for us, just remember the basal body. Fine. So here is the basal body. It is present below the plasma membrane. So here the plasma membrane would be here. Okay, and below the plasma membrane you find this basal body. It is like a microcylinder. It is a very small cylinder, so it is called microcylinder, which has structure similar to centriole. Actually, we will see it is a part of centriole. Okay, it is just a specialized type of centriole which is playing a part as basal body. Then you have rootlets. These parts that you see, they are striated fibers. Striated because can you see some stripe-like structure, right? So striated means striped. So they are striated fibers on outer and lower part of the basal body, and they are meant for providing support. So these actually provide support, and they will be present on the inner side of the plasma membrane. The rootlets are made up of bundles of microfilaments. So if somebody asks you what are these made up of, they are actually made up of microfilaments, right? So they are made up of actin, and they are not single actin, they are bundle of actin. Okay. Then you have basal plate. Basal plate is the structure which is present just above or just at the level of plasma membrane. From the basal plate, you can start seeing the new flagella, the outer part of the flagella or cilia forming. That outer part is called shaft. It is the hair-like projecting part with length of 5 to 20 micron in cilia and 100 to 200 micron in flagella. This shaft is covered by plasma membrane. Okay, so here is the difference. See, if you remember the prokaryotic this thing flagella, then you would recall that it had a basal body like structure. Okay, and then it had a hook, and then it had a filament. Now that filament was a single protein fiber. Okay, it was just a single protein fiber, and the protein is called flagellin protein. It was not covered by your cell membrane. However, in case of eukaryotic flagella and cilia, they are covered with plasma membrane. Okay, so there is plasma membrane present outside, and on the inside they are made up of many such fibers. Okay, they are made up of such many fibers, as we will see in the coming slides what these fibers are. But for now, if you take a cross section, can you see all these round structures? Okay, these circular structures are actually nothing but they are microtubules. Okay, so from this you can come to know that a prokaryotic cilia is just a single filament, while a eukaryotic cilia or flagella has such elaborate structures. Right. So now let us talk about the ultrastructure of flagella and cilia. This arrangement is called exonymy. Okay, so this exonymy is made up of what? It is made up of 9 peripheral doublet fibrils. Okay, one by one let us go through it. It is made up of 9 fibers. Okay, so here you have 1 2 3 4 5 till 9. So you can see that there are some 9 components present. What are they called? They're called peripheral. Peripheral because they're present in the periphery. Okay, on the outline, on the border. So peripheral on the sides, doublet fibrils. So if you see carefully, you can see one circle, and attached to that circle there is another circle, right? So this is actually 2 microtubules attached to one another. Okay, they're forming like double pipe like structure. That is why it is called doublet fibril. Doublet because it is double. Now it has doublet peripheral fibrils, but when you look at the center also has 2 single fibrils. Okay, these are 2 single fibrils. They are made up of tubulin protein. Just recall our microtubule session, and in microtubules I had told you it is made up of alpha tubulin and beta tubulin. So these all these fibrils are also made up of nothing else but microtubules. So you can say they are made up of tubulin protein. Okay, one more thing. These doublets, you can see they are attached to one another. However, in the center, what you see, these two microtubules are separate from each other. Okay, they are not joined like that. They are not joined like this peripheral fibers. This arrangement is called 9 plus 2 because you have 9 on the outside, okay, 9 doublet fibrils and 2 separate fibrils on the inside, right? So it is called 9 plus 2 arrangement. Each peripheral fibril, now let us consider or let us focus on this peripheral fibril. So each of them consists of a subfiber A. This whole thing, whole unit is called a fibril and a part of it is called subfiber. Okay, so this subfiber A is slightly narrower, as you can see from this diagram itself. The B part is longer or bigger actually, and A part is a little smaller. So it is slightly narrow and it has bent arms. Okay, we have already discussed it is made up of tubulin. Now what does A have? A has these bent arms. Can you see it has these two hands coming out of it, right? So it has its two arms made up of dynein protein, okay? And this has ATPase activity, which means if ATP is bound to it, it will have some other shape or form. When ATP is not bound to it, it will have some other shape or function, okay? We will see that later on. So this much is clear. 9 plus 2 arrangement. In 9 plus 2 arrangement, the outermost arrangement is called peripheral doublet fibers. In peripheral doublet fibers, you have the smaller or narrower one, which is called A, subfiber A, and you have attached subfiber B. On subfiber A, you have such dynein arms present, okay? Movement of flagella or cilia occurs because of sliding motion. So if somebody asks you how does a cilia or flagella move, it moves by sliding on one another. Okay, so just imagine these are nothing but these are tubes, right? So you have so many tubes all joined together. Now some of these tubes will move upwards. Okay, they will move up and some will move down. When that happens, okay, they slide on one another. When that sliding motion occurs, when one moves upward while the other one moves a little down, or the side which has moved up, okay, for example, the peripheral fibrils of this side 1 to 5 they moved up, and 6 to 9 they moved down. Then can you imagine which side will the cilia move? Very good, it will move on the right hand side. Are you able to imagine? So this is how by sliding it can move, okay? So sliding motion. In this sliding motion, the dynein arm establishes a temporary connection with subfiber B of the adjacent doublet fiber. So this dynein fibers that you are seeing, okay, these dynein arms will for some time, okay, bind to this B subfiber of the neighbor. So this A will bind to this B. That second one will also have an A. So that A subfiber will bind to next subfiber B, and in such a way they will be all connected for some time, and then they will release each other. Okay, so that is how this motion is occurring inside the flagella and cilia. The peripheral doublet fibers are interconnected by nexin protein. So this A and B, these subfibers, they are making this peripheral doublet fiber. So these fibrils, they are attached to each other by nexin protein. Okay, that protein is called nexin. Each A subfiber sends proteinaceous column to the center called spoke. So we can see this A, apart from being connected to B and to the adjacent fibrils, it is also connected to the center, and this connection is also by some protein fiber. These are called spokes. Have you seen in a bicycle there is a spoke, right? So same way, these are also called spokes. See, this is how it will happen. Can you see this? This is one subfiber A. It has dianin arms which are attaching and then detaching very fast. That is what is captured in this video. Okay, attachment and detachment very fast. Now let us talk about the motion. So flagella, it performs independent undulatory movements. First thing, flagella, they all move independently. They are not dependent on each other for their movement. Okay, every flagella can move on its own. It is free to move any way it wants. It has undulatory meaning wave-like motion. Okay, you can see it will move like a wave. While Cilia shows rowing type of motion. Rowing is like, remember that song, or if you have seen that row the boat, right? So how the oars they move in the boat, the same way cilia move in rowing type of motion. Undulations in flagella pass from tip to the base, pulling the cell through the water. Okay, these start from the tip. The waves they start from the tip and then they move along. Okay, and with this movement, the cell is able to move ahead. And this is the type of rowing motion you see in a cilia. Okay, can you see it is moving like that? The rate of ciliary and flagellar movements is 10 to 40 strokes per second. So this one whole cycle is called one stroke. Cilia and flagella can have movements in 10 to 40 strokes per second. Okay. Now here are the differences. As I had already mentioned, it would be better if you make these differences, okay, the tables for your own notes for quick revision. Fine. Just quickly going through all of these points which we just studied. The cilia, they have large number in a cell, 300 to 14,000. The flagella are only 1 to 4 in number. They are smaller in size, 5 to 20 micron in length, while they are longer in size, the flagella. They are 100 to 200 micron in length. Cilia, they occur throughout the surface of cell. Okay, they can be present on every surface of the cell. But flagella are normally found only on one end of the cell. Cilia, they can beat coordinated. Okay, so all the cilia will be moving in a rhythm. So this will be called synchronous. Okay, to synchronize. So from that comes synchronous. Or they can be what you can say, beating one after the other. Okay, like after one cilia or one region has already moved, then the next part will move. So in such a way, it can show rhythm or it cannot show rhythm, fine. But flagella, they beat independently. They do not care about other flagella. They just move on their own. Cilia produces a sweeping stroke. Okay, it is kind of a sweeping stroke or rowing motion, while flagella produces undulatory motion. Cilia help in locomotion, in aeration, in feeding, circulation, and other tasks, while flagella it only helps in locomotion. Cilia may fuse to form undulating membrane and surface is smooth. Okay, so just imagine if all of the cilia they fuse together. Okay, then they will form one smooth surface which can move. Fine. Fusion of flagella is unknown. No flagella fuse together. Okay, and the surface of flagella may be smooth or it may possess flamen. Flamen is just some spiky structure. Okay, however, it is not important for your NEET, so you do not need to remember. This is just for your general knowledge. Right, so the functions of cilia and flagella, they help in locomotion in flagellated and ciliated organism. They generate current by obtaining food from aquatic medium. So they can generate current. When they keep on beating, the water around them can move. And when the water moves, the food can come closer to them. Right? So in aquatic medium, to obtain food, they can generate current. Then also to renew oxygen supply. If near this organism, the oxygen levels have gone down, if the cilia and the flagella they keep moving, then new and new oxygen will be able to diffuse, okay, closer to this organism. So it is helping in renewal of oxygen supply and quick diffusion of carbon dioxide. In some protists and animals, they also take part in capturing food. Okay, just think about paramecium. Paramecium has so many cilia, and if you remember the structure of paramecium, all the cilia will beat in one direction, and it will allow the food to enter the paramecium's gullet. Okay, it has that groove. It has a small space through which food can enter. Let me just draw if I can properly. Okay, right. So this is how you would have seen a paramecium, right? And here it has this certain kind of groove. This is how you normally see. It has two nucleus, this one and this another one, and it has a lot of cilia on its body, right? So now what this cilia will do? Just think here. These cilia, and there is some food particle here. So these cilia will move in one direction and allow this food to move to be moved also in this one direction. Now, how does a paramecium eat food? This food has to enter this gullet. Okay, this part is called gullet, and it will enter from here and then it will be eaten by the paramecium. Then the canal system of porifera, that is sponges, has choanocytes which have flagella, and this helps in generating a water current. Okay, even sponges, when they create this water current from ostia, if you remember, which are the pores present on the side of the body, the water will move in and through the central cavity and the opening, which is called osculum, the water will move out, right? So this also happens with help of flagella. Then in coelentrates, they circulate food in the gastrovascular cavity. Like I had told you to imagine, think of a jellyfish, and the jellyfish has its gastrovascular cavity in which the food will move around because of cilia. In land animals, the cilia of respiratory tract help in eliminating dust particles coming from the air. So in your nose, okay, in your respiratory tract, you have the cilia. Ever wondered how mucus comes out of your nose? Right, so it is also moving because of your cilia, okay, the cilia present in your respiratory tract. Along with this mucus, the cilia will try to remove any dust particle, any microorganism which might be stuck inside your nasal cavity. Okay, so this is also a function seen by cells of respiratory tract, okay, ciliated. It is also important for internal transport in several organs. Okay, example, passing of eggs in oviduct. Now, if you just see the oviduct, this is the electron micrograph. You can see a lot of cilia present here, and this is just a cross section where also you are able to see a lot of cilia present above. Okay, this is the diagrammatical representation of the same. Now what happens is, when the egg is released from the ovary, this egg is to be transported to uterus, right? It has to move down the fallopian tube and into the uterus. The egg does not have flagella or cilia. So how will it move? So for its transport and for its movement, these cilia will beat in one motion and allow the egg to pass, or allow the ovum to pass from the ovary into the towards uterus. Okay, same type of cells, okay, ciliated cells are also found in kidneys, especially in the neck region of the nephrons. Okay, and they allow passage of excretory substances to generate a current within the nephron. Fine. Being protoplasmic structures, they also function as sensory organs. They can also act as sensory organs, especially think about ear. Okay, in when we will discuss ear, you will or if you remember ear, there are such ciliate hair-like structures in your ear, which are nothing but they are cilia. Okay, so these are functions of flagella and cilia. Now let us move on to the next organelle, which is centrosome and centriole. Okay, centriole is a membraneless microcylinder. Okay, it does not have a membrane. So if there is a question that which all organelles do not have a membrane, the answer will be centriole and ribosomes. They do not have a membrane. So centrioles, they do not have membrane. They are microcylinders. They are small cylinder-like structures with ability to form their own duplicates. They can form their own duplicates. Okay, they can replicate on their own. Here is what the centrioles look like. Fine, and you cannot see any membrane which would be covering it. It forms astral poles and basal bodies. Okay, the basal bodies of flagella and cilia, you remember? So these are actually made from centriole, and astral poles are the ones which are present during cell division. Okay, so during cell division, they form astral poles, and for locomotion or wherever you require a flagella or cilia, you have basal bodies which are also formed from centriole. They're visible under the light microscope. If you try and see, then this is the microscopic picture. You can sort of see where the centrosome will be present. It will be present here, okay, where the activity is showing more. Fine, so they are visible under light microscope. However, their exact structure is not visible, okay. For that, you require electron microscope. They are normally called diplosome. Okay, diplosome because two centrioles are present together. Okay, so these two centrioles are found together but at right angle to each other. So if you this here see this, okay, here, so there is one centriole and perpendicular to it there is another centriole, okay, at 90-degree angle. So they are at right angles to each other, and these two centrioles together, they are called diplosome. The complex of formed of centrioles and centrosome matrix is called centrosome. Now, whenever scientists looked at these centrioles or the diplosome, they found surrounding to it there is a very clear and a very distinct cytoplasm. Okay, surrounding these centrioles, there is a specific area of clear zone. Okay, there is a clear zone. So this area together along with the centrioles and this clear zone, it will be called centrosome. Okay, and this part of the cytoplasm is also known as centrosome matrix because it is a little different from the normal cytoplasm. Fine. Now centrioles are found in all eukaryotic animal cells. Okay, they are not present in plant cells. So the difference between animal cell and plant cell, you will be able to see that the centrioles are absent in the plant cells while they are present in the eukaryotic animal cells. They are also present in protozoan. Some protozoans do not have centrioles like amoeba. And then fungi, some fungi have. Then eukaryotic plants that have flagellated structures, okay, flagellated cells. For example, we were talking about those zoospores, or when we talk about spermatocytes which are moving around. Okay, so if they are moving around, that means they have flagella, and for flagella you require a centriole. So some eukaryotic plants which have such flagellated structures, they show centriole, but mostly the higher plants, okay, they do not have centriole. It is absent in angiosperms and gymnosperms. Fine. It is absent in the higher plants. Some algae also do not have centrioles, and instead they have something known as MTOC, microtubule organizing center. Okay, and this MTOC will help in cell division in case of plants. Okay, because they do not have centriole. Then you may be questioning that, okay, then how does this cell division happen in the plants? So it happens through this MTOC, which is microtubule organizing center. Centrioles are capable of replication. So this is one centrosome which has these two centrioles. It can replicate, okay? And when will it replicate? So its replication is coordinated with the cell division cycle. Okay, remember cell cycle? We will be discussing that chapter next. So in S phase, okay, during this S phase, synthesis phase, the centrosome or the centrioles will duplicate. So see, here it is happening. From two centrioles or one centrosome, you will now have two. Okay, and they will keep on separating throughout the cell cycle. Okay, like this. Centriole replication also occurs at the time of formation of basal bodies of cilia and flagella. So when the cell has to develop cilia and flagella, at that time also the basal bodies, they undergo replication. Now let us look at the centriole ultrastructure. Okay, how does a centriole look like if you cut it and you look at it under a microscope? So this is how it looks like under an electron microscope. Can you see how pretty it is? It has these microtubules, okay, and there are three microtubules arranged like this in a wheel shape. Okay, that is why it is called to have, or it is supposed to have this cartwheel type of appearance. Okay, like a wheel. Okay, centriole possesses a whole of 9 peripheral fibrils. Remember, in cilia and flagella also we had seen 9 peripheral fibrils. Here also there are 9 peripheral fibrils. But these fibrils, okay, each fibril is actually a triplet instead of a doublet. Okay, in cilia and flagella it is a doublet. In case of centriole it is a triplet. In center there is no fibril present. So it is called 9 plus 0 arrangement. Okay, there are 9 arrangements on the periphery, but in the center there is no arrangement. So it is 9 plus 0. Each fibril, this one whole thing is called one fibril. It is made up of 3 subfibers. Okay, you can see A, B and C. Right, from the outside to inside, if you want to name it, the outside one, the outermost is C, then B, then innermost is A. The adjacent triplet fibers are connected by CA proteinaceous linkers. So if you see here, these all ABC subfibers, okay, these each fibril is attached to the next one through an AC linker. AC linker, because if you notice carefully, it is linking the C subfiber of one pair or one triplet and A fiber of the second triplet. So this in between the bridge that you see is called AC proteinaceous linker. Linker because it is linking. Okay. In flagella and cilia, we saw these linkers were made up of nexin protein. Right? Fine. The center has rod-shaped proteinaceous mass. Okay. This is just some other proteins through which this hub is made. Okay, so the center is called hub and it is made up of proteins, fine, not microfibrils. Okay, some other proteins. From the hub, 9 proteins called spokes. Okay, such 9 spokes come out and they are also made up of proteins, and each spoke is connected to the A fiber of the triplet. Okay, so from these peripheral fibrils, the A subfiber will be connected to the hub through a spoke. So overall it gives it a cartwheel sort of appearance. Okay, this much will be clear to you. 9 plus 0 arrangement, there is no microtubule in the center. It is just presence of some other protein. Okay, not microtubules. Then this hub, this center, it does not have any microtubule. It is attached to the peripheral fibrils by spokes. Peripheral fibrils are triplet in nature and the triplets are C, B and A. Fine, and they are attached to each other through linkers. Now we discuss the functions of centriole. What is the function? They are capable of forming new centrioles. That is, they can duplicate their own self. Okay, so if somebody asks all the list of self-duplicating or self-replicating organelles, then you can write centriole in that list. Okay, then it helps in cell division and separation of chromosomes. As you can see here, these are the centrosomes or the centriole pair, right? The centriole pair is connected to these chromosomes, okay, through microfibers or microtubules or through spindle fibers, right? So these spindle fibers, they are attached to the chromosome and they will help in cell division and separation of the chromosomes. Then they form the axial filament or the tail in sperm. So this here is a close-up view of a sperm, where this is head, where there will be nucleus. Okay, then there is a neck region, if you know. So in this neck region there are these centrioles. Okay, two centrioles, one is present facing us and one is present perpendicular to it. Fine. So this one is called proximal because it is in proximity, okay, closeness to the nucleus. And the second one is called distal, just to identify each of them, right? So here is where the centrioles are present, and through this distal centriole, actually, you will see that there are fibers coming out, and they will form the single flagella, okay, or the tail in the sperm. Centrioles can be transformed into basal bodies. Okay, so here you have a centriole pair, and from this centriole pair you can see that one of the centrioles has taken up the process and changed itself to become a basal body for a cilia, right? Now this centriole will act as a basal body and it will synthesize this cilia, fine, giving rise to cilia and flagella. This is how it will look in a cell, and if there is a ciliated epithelium, there will be a lot of such basal bodies attached at the bottom, and through these there will be cilia or flagella coming out. So these are the functions of centriole. Now can you see the resemblance between centriole, cilia and flagella? Yes, right? So because all of them are made up of microtubules, right? They are all made of microtubules. All of them possess 9 peripheral fibrils, right? When you look at the number, then both of them have 9 peripheral fibrils. The difference is that in centriole, you can see these are the centrioles. How can you recognize? Because they do not have central fibrils, okay? They just have side fibrils, peripheral fibrils. So 9 plus 0 arrangement in centrioles and 9 plus 2 arrangement in case of cilia. In case of yes, cilia. 9 plus 2 in case of cilia and flagella. Fine, which is shown here. The 9 in the periphery and 2 in the center. Basal granule present at the base of cilia and flagella is derived from centriole, as we just saw. All three of them are capable of movement. The centrioles, they move inside the cell. Okay, it is limited to move inside the cell only, while the flagella and cilia, they can produce current and they can cause locomotion in the organism. They will help in feeding, they will help in aeration, a lot of functions. They are very movable and flexible parts of the cell. Fine, right. Now we come to the last part of today's lecture, which are the microbodies. Okay, these microbodies are, as the name suggests, they are small. Okay, small organelles. They are not seen in all of the cells, and they have very specific functions. So they are seen only in some eukaryotic cells. We shall see. The first one. Okay, well, let us just first discuss what microbodies are. So they are present in the cytoplasm. So they are also known as cytosomes. Okay, they are small organelles present inside the cytoplasm. So they are called cytosomes. They are very small in size, 0.2 to 1.5 micron, and can be seen with electron microscope. Some of the common characteristics of all microbodies that we will be discussing are: they are vesicular and mostly spherical in shape. Okay, they are vesicle-like and spherical in shape. Enclosed in a single plasma membrane. Okay, they have this single lipid bilayer membrane. Okay, the matrix, which is the inner portion. Okay, the vesicle is filled with something. So what is it filled with? That filled material is called matrix, and it is filled with, or it contains, proteins and enzymes. Okay, let us start with the first microbody, which is sphaerosomes. Okay, sphaerosomes, they are small cell organelles bound by single membrane and take part in storage and synthesis of lipids. What is their function? They are storing and synthesizing lipids. Fine. inside a cell. So this is how they look like in groundnut. Groundnut, you must have known, you even get groundnut oil, right? So where is this groundnut oil coming from? Of course, from the seeds. Where in the seeds? Of course, from the endosperm. Where in the endosperm? So endosperm cells have such sphaerosomes in abundance, and these sphaerosomes are the subcellular particles or organelles which contain and store lipids. Clear? It is 0.5 to 10 micron in diameter. It arises from the endoplasmic reticulum. Okay, where does it come from? It comes from endoplasmic reticulum. Its biochemical composition is 98% lipid. It is mostly made up of lipid, and then there are some proteins also which are found on the membrane, as well as some which are found inside. These proteins are, first thing as I told you, they are present on its membrane, and secondly, they are either enzymes for fatty acid or fat synthesis. Okay, so for fat biosynthesis, there can be enzymes present in the spherosomes. Because of presence of lipids, spherosomes can be seen under light microscope after staining. You normally stain it with dyes like Sudan black, okay? And when you look at it under a microscope, so this is the whole cell, but it is storing a lot of oil. So these oils will be present in what you see, this granular structure. Each of these granular structure is actually a spherosome storing lipid. It is abundant in endosperm of oil seeds. Oil seeds are the seeds which you use to extract oil, okay, like groundnut. Spherosomes of some tissue contain hydrolytic enzymes, thus they have some lysosomic activity. They are little bit like lysosomes in some conditions because they have some hydrolytic enzymes. They are also known as oleosomes. Okay, their other name is oleosome. The second microbody is called peroxisomes. Peroxisomes, as you can understand, they take part in peroxide biosynthesis. Okay, what is peroxide? Hydrogen peroxide you have heard of, H2O2, right? So wherever you have this O2 minus ion present, it is called a peroxide. I think in chemistry you will be studying this peroxide ion, superoxide, oxide, right? So this is a peroxide, and peroxides are synthesized inside the peroxisomes, and which gives it its specific name, right? How does it look like? It has a lipid bilayer, so a single membrane. Fine. Inside the single membrane, there will be matrix, and there will be a crystalline core. This core or crystal-like core is actually made up of proteins. Okay, it is made up of proteins. Peroxisomes are found in all eukaryotic cells, but they are generally in close association with the endoplasmic reticulum, mitochondria, and chloroplast. Fine, because along with endoplasmic reticulum and mitochondria and chloroplast, they will be performing metabolic activities. So they are found closer to these organelles. They are quite abundant in liver and kidney. Liver, as you already know, it does the function of detoxification. Okay, it cleans your blood. So whatever particles or whatever drugs it has to metabolize, it requires peroxisomes. So it is abundant in liver and kidney. A photosynthetic cell may have up to 70 to 100 peroxisomes. Okay, so in plants, you can have 70 to 100 peroxisomes. In a photosynthetic, which means a green cell. Okay, in a chlorophytic cell. Peroxisomes are also believed to develop from endoplasmic reticulum. Their size and shape are variable. Okay, you can find them, some of them which are larger than small mitochondria, and other peroxisomes can be very small in size, also smaller than the mitochondria. They contain oxidative enzymes. Okay, they have such oxidative enzymes like urate. Okay, I forgot U here. It's not rate, it's urate. Urate oxidase. then D amino acid oxidase, alpha hydroxy acid oxidase, beta hydroxy acid oxidase. Okay, so it has all of these. This is the diagram where you can see it is doing so many metabolisms. Okay, so in metabolic function it is required. The reactions produce H2O2, hydrogen peroxide. That is its function. Okay, to produce hydrogen peroxide. And with help of this hydrogen peroxide, it will oxidize. Hence, it is said oxidative enzymes, right? And this hydrogen peroxide, so that it does not harm the cell, it is directly converted into molecular oxygen, O2, by another enzyme found in peroxisome called catalase. Clear? In animal cells, peroxisomes metabolize number of toxic substances. Okay, things that are toxic for you are cleared or they are metabolized by peroxisomes in your liver. Fine. So they metabolize toxic substance like nitrites, phenols, formaldehyde, formic acid, methanol, ethanol, etc. So in people who are chronic alcoholic, okay, they drink a lot of alcohol, their liver, and specifically in liver, the peroxisome can clear these alcohols. At least 40 or 50% of these alcohols are cleared by peroxisomes. Then uric acid. Uric acid also you must have heard of. There are three types of animals, right? The animals which excrete urea, then animals which excrete ammonia, and animals which excrete uric acid. So this uric acid is actually a breakdown product of your nucleotides. When your ATGC, they break down, okay, when they are degraded, they form uric acid. So it is nucleic acid catabolism product, and this uric acid is changed into allantoin inside the peroxisome. Okay, if you have studied then the snails and some smaller organism, they excrete in form of allantoin. In root nodules, they convert fixed nitrogen into urides for transport. When we will study this transport chapter, no, not transport, this mineral nutrition chapter. In mineral nutrition, we will also discuss nitrogen fixation and nitrogen cycle. So in this fixation, there is a part which says that the nitrogen, it is transported inside the plant in form of either amino acids or urides or allantoin or any other such molecules, okay, nitrogen rich molecules. So in root nodules, where the bacteria are found, where there is symbiotic relationship, this this fixed nitrogen is converted into urides. And finally, in photorespiration, which we will see in photosynthesis class, so there is photorespiration where the oxygen binds to the ribulose 1,5-bisphosphate, and this whole cycle is completed in three organelles, okay, in chloroplast, in peroxisome, and mitochondria. So this is also how peroxisomes help in the metabolism and photorespiration. The last type of microbody are glyoxysomes. Glyoxysomes, as the name suggests, it performs the glyoxylate pathway. Okay, from that molecule, its name is called glyoxysomes. They are microbodies which contain enzyme for beta oxidation of fatty acid. I had told you when we were discussing mitochondria that fatty acid oxidation occurs inside the mitochondria, and it is called beta oxidation, right? So fatty acids are broken down inside the mitochondria, while they can also be broken down into the glyoxysomes here. Okay, so in mitochondria also it can happen. It can also happen inside the glyoxysome. They are considered to be special peroxisomes. Okay, they are very much similar to peroxisomes. The only difference is they perform this glyoxylate pathway, and they are seen only for transient period. Okay, they have a single covering membrane and enzyme-rich matrix which has a crystalloid core, same as peroxisome. Okay, this is how a glyoxysome will look like. They appear transiently in germinating oil seeds. So if you germinate or keep any oil seed for germination. As the name suggests, it is an oil seed, which means it's endosperm. Okay, the food it stores is in form of oil. Now, this oil is not very useful when you have to produce a whole new organism. You require sugar, you require other molecules. So, what will happen is in this germinating oil seeds, the oil will be converted, the fatty acids will be converted into glucose. So, that cycle takes place inside glyoxysomes and it is called glyoxylate pathway. Remember, this is very, very special for germinating oil seeds. Humans cannot perform glyoxylate pathway. So if you eat a lot of oil, it will not get converted into glucose inside your body. Okay, that does not happen. Fine. They reappear in senescent plant tissues. Senescent is almost towards death, okay, aging plant tissues. For degradation of lipids, again they degrade the lipids and mobilize the products, okay, they mobilize these smaller molecules which are obtained after degrading the lipids. This is the glyoxylate pathway. Not very important for you to remember every step or anything, just for your information. Fine. This is the normal TCA cycle. Can you see? Citrate, isocitrate, succinate, alpha ketoglutarate, etc. etc. So this will be your TCA cycle. What the glyoxysomes do is this TCA cycle will run by fatty acid oxidation also. But when it reaches this stage from succinate, it forms glyoxylate also. So succinate plus glyoxylate, it will give from isocitrate, okay, and this glyoxylate will fuse with one more acetyl CoA, fine, to produce malate. Now this isocitrate is 6 carbon, succinate is 4 carbon. So it is giving you succinate which is 4 carbon plus glyoxylate which is 2 carbon. So 2 carbon plus acetyl CoA 2 carbon will give you malate. Malate with help of malate dehydrogenase enzyme becomes oxaloacetate, and this oxaloacetate it will undergo gluconeogenesis, okay, to form glucose. So this is how glyoxysomes are important for the cells or the plant cells and the seeds which store fat as storage of food. Okay, so here we end today's lecture. In the next lecture, we will be ending the chapter cell with the last part, which is nucleus and the cell, other cell organelles, especially. We will also be talking about the chromosomes, which are found inside the nucleus. Okay, see you next time.